in meiosis, half of the chromosomes are going to be given. So this set of chromosomes and this set of chromosomes, boom, are going to be given to the offspring. And then half of these chromosomes are given to the offspring. And so there you go, right there. So with that, 28 chromosomes, 14 were given. So let's put a 14 on this. 14 were given in a gamete. Of 14 chromosomes, 7 were given in a gamete, because that's what you know about meiosis, right? So this new hybrid has 14 of mom's chromosomes and 7 of dad's chromosomes. So it has 21 chromosomes, but this hybrid is sterile. So this hybrid being sterile, it's still able to self-fertilize, okay? So self-fertilizing can take place, and when it does that, it is going to go ahead and through a cell division error, meaning it didn't divide like it was supposed to, the previous example I was trying to show you, and self-fertilization, it can also end up with doubling everything. So you have A, B, and D chromosomes. So now you have double of the A, double of the B, and double of the D. Boom. So 21 chromosomes then doubled and became 42 chromosomes. This because it has that, this had an odd number of chromosomes, this has an even number of chromosomes, it can then in turn be fertile, and unlike its parent that was sterile. So, okay, so I hope that helps a little bit with that alloploidy, meaning that it has more chromosomes than its parent. Let's go ahead and clear this. You can find this on your D2L. And go ahead and go back to your PowerPoint. And basically this was trying to do the same thing. I just wasn't digging it. Let's move on. Alrighty, so then in polyploidy, most of your crops, your cotton, your wheat, your cotton, your oats, wheat, potatoes, and tobacco, they are polyploids. I just showed you the example of the wheat. And so your plant geneticists, they can produce new polyploids, and they can use chemicals to induce errors in cell division in order to do that. So they can produce more species. All right, so next, sexual selection. Sexual selection can drive sympatric speciation. So your sexual selection for mates of different colors has likely contributed to the speciation of cichlid fish in Lake Victoria. And so remember, your sympatric speciation means there was not a geographical barrier, okay? It means that they had speciation take place in the regular population. Speciation just took place. And so the next thing is habitat differentiation. Habitat differentiation can also result from the appearance of new ecological niches. So the example that they give are the populations of the North American maggot fly. They can live on native hawthorn trees, as well as more recently introduced apple trees. And so the flies that use the different host species experience both habitat and temporal isolation. So if they live on different trees, even though those trees might be near each other, they can actually speciate differently. They could literally be right next to each other, but they are indeed considered new ecological niches. So a review of your allopatric and your sympatric speciation. In allopatric speciation, geographic isolation is going to restrict gene flow between the populations. Reproductive isolation could then arise. If gene flow is not happening and you end up staying separate long enough, your population stays separate long enough, then eventually, even if you tried to get together, your genetic makeup could no longer be matchable. That's a word. So Even if contact is restored between populations, interbreeding is prevented. So that's how you know you have a situation of allopatric speciation. In sympatric speciation, a reproductive barrier isolates a subset of populations without geographic separation. It just happens literally within their population. And that sympatric speciation results from either polyploid, natural selection, or sexual selection. So the next thing to look at is hybrid zones. So your hybrid zones are going to reveal factors that cause reproductive isolation. What a hybrid zone is, it's an area where members of different species can mate and produce hybrids. The hybrids are going to be the result of mating between species with incomplete reproductive barriers, meaning they don't quite have the barrier, but are they a different species is the question. So a hybrid zone can occur in a single band where different species meet, okay, where they could actually have crossover in their niches and meet. And so the example that they give are two species of toads. They're in the same genus and they interbreed in a long and narrow hybrid zone. And so we're about to look at the zones and it's going to come into play. You're going to find a lot of times, though, the hybrids have a reduced fitness compared to the parent species. They have a reduced fitness because, like I was telling you before, they don't necessarily have they have instincts for one way, and they also have instincts for another way, or you might find that due to their physiology, their morphology, the morphology, they maybe should have long legs, but turns out they have short legs. I've seen this. I had a Labrador retriever with short legs. Why? Because it was a hybrid. It was basically a mixed breed, a hybrid of that and what was she? I can't even think what it's called. Cocker Spaniel. She was half Cocker Spaniel, half Labrador retriever. And so she looked like a lab, but she was about as tall as a tall Cocker Spaniel. And so that could reduce their fitness. So look in here. Here are your toads. And you can see in the orange, you can see the fire-bellied toad range here. And in the yellow, you have your yellow-bellied toad range. All along the area where that meets, you can find yourself what is called a hybrid zone, where those toads are having babies with each other. And so if you look here at your little chart, you can see distance from the hybrid zone center. So the further, so distance 40 kilometers, 30, 20, 10, 10, getting close to that zone. And then you go on this way, distance gets more again. And then what are we looking at? The frequency of the alleles between the variegata. So this one is the yellow-bellied alleles. And so the frequency in the alleles, you're going to find the further the distance from the hybrid zone, this is going to be the frequency of alleles. So as you get closer, you're going to have more individuals with more frequency of alleles. Then as you go through the hybrid zone, those frequency of alleles is going to go down. And then as you cross over into the other species, the frequency of alleles goes down even more. So with that, you're going to find healthier populations and more frequency alleles, in this case, in the yellow-bellied, in their deep zones away from the hybrid zones. But in the case of the fire-belly one, as you're looking here, you're going to find that they are better as hybrids than they are on their own. Pretty interesting. All righty, so hybrid zones over time. So if hybrids don't become reproductively isolated from their parent species, there's 3 different alternative outcomes, okay? Reinforcement, fusion, and stability. So what does that look like? So if you look here, there's your three possible outcomes: reinforcement, fusion, and stability. So to look at this little diagram, you have an original population, okay? So gene flow between those, between the populations. So you can have population, 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 they're neighboring, you got gene flow going between them. Okay, and so then now you have these two populations with gene flow between them, and now you have a barrier. So the individuals on this one here, there was a barrier where gene flow could not go between the populations. So then they became reproductively isolated. And so when you look over here, they started changing over time. They started changing over time. They started changing over time. Okay, stay there, we'll come back. So these two populations, they continue gene flow between them. By continuing gene flow between them, that's continued to allow them to be healthy populations. And there wasn't a lot of changing over time because you have that constant gene flow between them. What made these change over time was the fact that they were reproductively isolated from other populations. So this, what you're seeing here, is a hybrid zone. Okay, so this hybrid zone is when individuals from this population, and we're talking way later in time, get together from individuals from this isolated population, they get together from there. So the question is, of these three possible outcomes, reinforcement, fusion, and stability, those are three possibilities if these guys get together. So let's see what those look like. So when hybrids are less fit than their parent species, reinforcement is going to take place. So basically that's reinforcement of those reproductive barriers. So I wish I had a big one. I don't. So when you look over here and you see this population goes in this direction and evolves, and this population goes in its direction genetically, okay? You may have your hybrids in between, and you may have some individuals being successful, but as you notice, it's a small amount, okay? So the most successful individuals are going to be those of your isolated populations. Those populations of the original populations are going to be the strongest of those populations. And that's what reinforcement means. So over time, the rate of that hybridization gets smaller and smaller and smaller. And so where reinforcement occurs, reproductive barriers should be stronger for that sympatric than allopatric species. And so I'm gonna let you read about this. I really want to go cover the next piece. And so if hybrids are as fit as the parents, okay, and there's enough gene flow between them, if that gene flow is great enough, those reproductive barriers are actually going to weaken, and the parent species can fuse into a new species. So if you have that population that got isolated, and then these other populations, and they came together within their hybrid zone, the next one is called fusion. Where did it go? I guess it didn't really say it, but it is fusion. So you have one population and then another population around the hybrid zone. Those hybrids are very successful. They're just as successful as their parents, and they can reproduce with each of those parents. So the hybrids can reproduce with each other. The hybrids can produce with the first peak parents, and they can reproduce with the purple parents. So that will in turn eventually fuse them into one species. So that's called fusion. So the next one, let's make sure it's going to go ahead and find in the stability. Yes. All right, so extension gene flow from outside the hybrid zone can overwhelm some selection for increased reproductive isolation inside the hybrid zone. So in this situation, in stability, you are going to find You have your original parent population sets and these population sets, and your hybrids are going to be in between. So that's going to find yourself in a situation of stability. Okay. In that situation of stability, population A, parent population A, is successful and does well. Parent population B is successful and does well. The hybrids in between are successful and does well. So population A and B can continue to hybridize, but the hybrids are also equally successful. So you will find a situation in stability within that hybrid zone. So the first one, not successful, less hybrids. The middle one, successful, but so close that eventually they're going to end up as one species. And then stability, staying separate, and all of them are being successful. So how long does it take? Questions on how long does speciation take to form? How many genes need to be different? And so that rate of speciation can be studied using that fossil record, morphological data, and molecular data. Molecular data right now is really, really, really what is helping out. When we're using the fossil record, remember I told you before that we're really dealing with organisms higher than the species level. And so the interval between speciation events can range between 4,000 years, between some of those cichlids fish, it's a weird name, to 40 million years, like some beetles, with an average of 6.5 million years. So 40 million years to, yeah, nice average. So the fossil record includes examples of some species that appeared suddenly. Okay, and they persisted unchanged for some time, and then they disappeared. So the term that you should know, punctuated equilibria, it describes these periods of apparent stasis punctuated by sudden change, where everything was the same, then all of a sudden there was a change. That's what that means. So the punctuated equilibrium model will contrast with a gradual model. And so if you look at the two models, punctuated model, sometimes you'll hear it as punctuated equilibrium versus the gradual model. Follow time. So this is situation A up here, and this is situation B down here. Time goes in this direction. In the case of the punctuated equilibrium model, you have one species, and then there's such a big genetic mutation in the offspring of that species. Now you have different species. And then throughout time, they maintain, just like such. But there was a big punctuation right there where there was a big change. The gradual model of speciation, the tempo, basically, if you look at this butterfly and you can see the offspring still similar, still close enough to be called same species. This offspring, its lineage goes and changes over time. Like I said, what does it say? 6.5 million years? Sounds like a good average. And then now you have new species. While this offspring changed over time, average 6.5 million years, and then you have a new species. So this gradually took on mutations and retain those mutations. This one, quick mutation. So in studying the genetics of speciation, the fundamental question of evolutionary biology persists. How many genes influence the formation of a new species? Well, it really depends on the species in question, okay? Speciation might occur, might require change in a single gene, and it makes it so reproductively isolated in just that single gene, doesn't matter. Or it could take the adding up of several mutations over the course of millions of years in order to become that new species. And so your example is, they give an example of your Japanese Euhadrus snails. So the direction of the shell spiral affects mating. okay? And it's controlled by a single gene. So if the shell, let's see if I can get this to, I sure can't. What's going on here? There we go. Let's draw my little pin color. If one snail, his shell spirals in this direction, right? But a different snail, his spirals in this direction. Doesn't seem like a big deal, but if it keeps their parts from meeting up, then it's a big deal. So in the monkey flowers, at least 2 loci, so 2 different parts on a chromosome, will affect flower color. And that flower color influences pollinators. So different pollinators are going to like different colors. And so that could make a big difference, just the change in the color of the flower. So pollination is going to be dominated by either hummingbirds or bees and could lead to reproductive isolation of those flowers. And so in other organisms, speciation can be influenced by larger number of genes and gene interactions too. So not just one gene, but groups of genes that need to interact with each other. So macroevolution. Macroevolution is going to be the cumulative effect of many speciation and extinction events. So macroevolution, remember, is on a large scale. It is going to be the big picture versus microevolution is the alleles, the little picture.